similar question last time. It was the LTS, like discrete signals one. And I'm having trouble like differentiating between the this question. I think it's talking about the same thing. It's talking about number of signals,
Mhm.
and it's talking about this compound right here. But it's saying like to aromatic hydrogen atoms. Like what does that mean? Is it talking about each one of these?
Yeah, it's talking about the ones that are part of the ring,
Okay.
right?
So then how do I know which ones like how do I know how many signals there are?
So you'll get a signal for, each non-equivalent neighbor. So for instance, this one is bound to this carbon, which is bound to this carbon and this one. Okay?
Okay.
And in blue, I would say that this carbon, maybe I'll do it like this. So this carbon with its hydrogen is bound to this carbon, which is part of this ring here. And it's also bound to this carbon, which is just bound to that hydrogen. In blue, I
Okay.
would then put for this carbon here, its hydrogen is also bound to this hydrogen, just like this one, and this one.
Yeah.
So that means that this and this are equivalent.
Since they're bonded to the same thing basically.
Yeah.
So this one's bonded, they both share this right here.
Yep.
And then they all share this one right here.
Yeah.
Okay.
Now, the HA and the HD, the reason that these are the same is because if you consider it, I'll use green, like this carbon, you have what's next to it is a carbon bound to an H, and then what's next to it is also a carbon bound to an NO2, right?
Yeah.
And then same thing here, I'll use orange. So this one is bound to a carbon that's just bound to a hydrogen like this, and it's bound to this carbon that's bound to the nitrite.
So isn't that another signal?
Yep. So you'll get
But
your,
then
yeah.
so these only count as one on this side, and then this count as one because they're the same.
Yeah.
So it's not four different. Oh,
Mhm.
okay. And then, so like if we were to compare it to the one we did the other day, looking at it says how many discrete signals are present in the, I guess they're talking about the whole thing here.
Uh-huh.
But this one they have three. So let's say look at this carbon right here. It's connected to this,
There's no carbon there.
or
So you could
mm,
have a carbon here. I mean, sorry, a hydrogen there, here, here, and here, as well as,
yeah.
these, right? So
Hmm.
it actually is just like that other one because you actually have this is equivalent to this.
Yeah.
And I'll use green. This is equivalent to this, right?
Yeah.
Because for this one, it's bound to this carbon that's bound to the sulfur or whatever. And it's also bound to this carbon, which is bound to just a hydrogen. And then this one over here is bound to a carbon that's bound to the sulfur, and it's also bound to the carbon that's bound to this hydrogen. So this and this are equivalent. And now
So
for,
that's one.
yep. And now for this one, this hydrogen is bound to a carbon with this hydrogen, and it's also bound to the carbon that's bound to the NH2. And same thing for this one. So that's why these ones in green are equivalent as well.
Yeah.
So, uh, and now lastly, we have these hydrogens, the ones in the amine, and
Mhm.
those have no neighboring hydrogens.
Oh, okay.
So, so
So
these,
these two are equivalent. That's one. These two are equivalent, and this one by itself has no equivalent, but it still counts as a discrete signal.
yeah, basically the blue hydrogens have two uh neighbors. So
Mhm.
that'll give you your triplet, and same thing for this one. And then for this one, you'll just get your singlet.
Which one gives you the signal? The singlet?
The one
Yeah.
that has no neighboring hydrogens.
So this one right here would be a
Yeah,
singlet.
yeah.
Yeah. Okay. So and then right here. So this is bounded to the one. That's one, and then this one.
Mhm.
Oh, okay. All right. Okay, that makes sense. Okay.
What if this said NH2?
Then it will be three signals because
Yep.
this one's by itself.
Mhm.
It would be a singlet. So these two would be the same.
Yep.
These two would be the same, and then this one would be by itself.
Good.
Okay. All right. That makes sense. Okay. Okay. I had a question on the difference between like aerobic and anaerobic metabolism,
Okay.
and then like how that kind of relates to like the electron transport chain. And
Yep.
I'm having trouble like picturing all that. Like what happens if you like turn this off or like if you don't have enough of this. I kind of just guessed on this question. I don't really know.
All right.
Like it says which of the following would like to
What
decrease ethanol.
Okay. So, uh, so aerobic means with oxygen,
Mhm.
and anaerobic means without oxygen. Okay. So anaerobic examples would be glycolysis and the lactic acid cycle, which for yeast is uh alcohol fermentation. So those things are anaerobic, whereas like the Krebs cycle and electron transport chain, those are aerobic with oxygen
Oh,
present.
so glycolysis, lactic acid, and then alcohol fermentation are anaerobic?
Yep.
Okay.
Because yeah,
And then the Krebs cycle and the electron are
are
the
aerobic.
okay. And the
Basically,
TCA cycle?
yeah, that's the same thing.
Yeah, so same thing, yeah. Okay.
So the main thing is oxygen.
Hmm. So
So,
like if you go through this question right here
mhm,
and it says which of the following would lead to decreased ethanol production, we're talking about the the alcohol fermentation.
mhm,
So then if you decrease it, that means you have it's something to do with oxygen.
okay.
But then I'm not sure how it's like.
So it
Oh, wait. So if there's no oxygen, there's less yeast production.
uh, uh if there so yeast ethanol fermentation is anaerobic, right?
Yeah.
So they're asking an increase in which of the following will lead to a decrease. So oxygen is inversely proportional to the fermentation, right?
Hmm.
So an increase in partial pressure of oxygen, which will be the oxygen concentration, would cause a decreased production of ethanol, right?
Yeah.
So,
Based on this, yeah.
yeah.
Okay. And then why are the other ones kind of wrong?
So pyruvate kinase is what
It's
turns
for
the PEP into pyruvate, and if you increase that, I mean, so that's part of glycolysis. If you increase
yeah,
glycolysis is anaerobic. So that won't cause a decrease in ethanol production.
it would increase it.
And then C,
And then
concentration of glucose. If you have a high amount of glucose, it just means that you're gonna have, more of that glycolysis glucose breakdown. So, that wouldn't work either. And then Krebs cycle inhibitors, that's inhibiting the Krebs cycle. Now um we know that Krebs cycle is aerobic. So if you have an
mhm,
increase in the Krebs cycle, you would have a decrease in ethanol production. But D is saying Krebs cycle inhibitor. If you inhibit the Krebs cycle, then you should
you would
have
have more ethanol.
correct.
Okay. Yeah.
You see how
Okay.
a proportion like this helps
Yeah.
us answer this type of question without us
Hmm.
being confused. So you always want to identify these types of relationships.
Yeah. I didn't really know that from like relationship.
Yep, yep.
I
Proportion.
know like the insulin and glucagon one, but like this one I was like, don't really know.
Oh yeah, yeah. Um, yeah. So,
Okay.
hmm,
And then just had one last question about the voltages and circuits. Like I know like the formulas of like resistors and like series and then parallel, but I'm having trouble like how do I apply it to certain like questions?
um, so I mean, this is going to be like significantly harder than when you see on the exam, but we could still do it. And yeah, one of the reasons that it's significantly harder is because you have what is it called? Two sources of voltage,
Hmm.
like two voltage sources. So let's see. We have How, okay. How would you,
Well, I was looking at these two first. And I was like, so these are like, I feel like they're in series. So I'd add these two up, and then this one's in parallel. So I would just add these and then do the inverse of this. But then these voltages kind of like threw me off.
so, so they seem to come from like voltage one, electricity comes from that and goes through resistor one. Voltage two, electricity goes into resistor two, and they pass through the resistors and then they meet at point A when they combine to go through resistor three. And then I believe they'll split again. So let's see. Um, okay, so I'll call this current one, current two, and then of course current three. And it should follow that current one plus current two is equal to current three, right?
Yeah.
Okay. Now that's the okay, the junction rule. And also we have Kirchhoff's loop rule in which the net voltage around a loop in a circle is equal to zero. Right? So they are okay. Let's see. The okay, so the resistors or okay, each voltage source is 10 volts and the resistance of each resistor is 5 ohms. What is the current exiting point A into R3? So,
Wouldn't you just do like V equals IR and then try to solve for the current I equals V over R,
mhm.
and then I don't know, combine these voltages and add up these resistors?
Oh, these will. both be 2 amps, but that wouldn't work because then I would have 4 amps, which obviously won't work. So let's see. Okay, so at point A, okay, voltage at point A can write as okay, let me try this. R1 and R2 should be in series, give us 10 ohms, and they are in parallel with R3. So let's call this R12, and then R3 should be um, oh, okay. Um, Hmm. I'll come back to this one.
Okay.
Yeah. Okay. In the circuit shown, a current of 2 amps flows through the 40-volt battery. What is the value of the unknown resistance R? Um, okay. Let's see. So when we have a junction, that's how we know that we're splitting something into to make a them parallel.
Mhm.
So let's see. Okay, let's see. So these combine to be 30. These also combine to be 30. Um, oh, okay. I got it. So
Okay.
basically, okay, so we have this unknown resistor, and then we have a junction here, which means that the current is going to split, and these will be in parallel with these. However, oops, like these two are in series, right?
Yeah.
So if they're in series, we can just add them, right?
Okay.
So that would be combined to give you 30 ohms. And same thing with these two 15 ohms resistors, right? We could just,
Hmm.
they're in series, we could just combine them by adding them. So we also get 30 ohms as the equivalent resistance, right?
Yeah.
So now that's the same thing then as if we did this with, oh, so to add these two in parallel, we do
One.
1 over REQ is equal to 1 over 30 plus 1 over 30, which is 2 over 30, which is 1 over 15. So the REQ is 15. So it's like 15 ohms here, right? Voltage of 40 volts. Current is 2 amps. So and then this is the unknown resistor. So then I just basically did V equals IR to get R um isolated, and R is just R plus 15 since R is in series with this
Wait,
15.
can you explain that part again?
Uh
Like how you get to the R plus 15?
so does it
Like
make
this part makes sense. So you got the 15 ohms here.
huh?
So, and then you have the resistance.
Yeah.
So, okay, this part I understand. Like we're up to here. But then how do you do the calculation to like get that?
So so does it make sense that this is in series with this?
Yeah. So this is just the calculations from this.
Yeah.
Okay.
So yeah.
Oh, okay, okay. So you're adding these two. So 15 plus this because they're in series. V equals I. V equals I. So it's R plus 15 equals V over I. Oh, okay, okay. So 40 over 2 is 20. Oh, okay, okay. I see that. That makes sense.
Mhm.
Okay. Okay, that makes sense. I just didn't know that these two were also in series of each other, so you can just add them.
Yeah, so it can so basically in order for something to be parallel, you need a junction like this, right? But a lot of times these can be drawn in ways that make it hard to see. So that's why I
Mhm.
redrew it like this over here.
Yeah, this makes more sense than this.
Hmm.
Okay. All
Yeah.
right. I was thinking of maybe we can go through like the new section bank
Huh?
questions, and maybe like I go through each like
Yep.
maybe I do a passage, and then you can give me like
Yep.
tips on like maybe how to review it or like
Yep. Let's do it.
certain stuff. Because I haven't seen these at all. And, oh wait, let me just.
Yeah, me neither. I was thinking of doing like a live stream of me like answering, like doing it for the first time.
Oh, okay. Yeah, I think I heard you talking doing something
Yeah.
like that. Oh wait, why is it not showing the Jack Weston so now this one opens with Jack Weston. Why doesn't the other one?
Um,
Do you know? Or maybe it's not updated or something.
yeah, they probably because it's new.
Oh, okay.
Because
So we're
the
just gonna
because the yeah. So it's because it's likely new, so they haven't gotten to writing out the solutions.
okay. All
Yeah.
right. Sounds good. All right. An adult individual experiencing diarrhea complained of weight loss despite consuming a large amount of food. Researchers took a biopsy of the pancreas and small intestine. I usually highlight like certain body parts, and then collected a stool sample and performed a glucose tolerant test where glucose levels are tested under two conditions. One's after fasting, and then after 2 hours of consuming 75 grams of glucose and 237 ml solution. These were the results. Researchers detected glucose in the individual's urine.
So why don't you tell me what's happening, you know, in this?
So
So
they're doing a test to check their blood glucose after eating food because the individual was experiencing diarrhea and they want to see their glucose levels when they're fasting and then after eating.
yeah, so we could see that um for fasting, the glucose level was less than 95 for the standard value. Wait, I'm not sure what the standard value versus
Is this
result.
what's like normal?
Oh, and then the result. I see, I see.
And
Okay.
this is the results.
So it should be less than 95, but it's actually 135. So it's a high fasting glucose. And then after 2 hours of consuming, glucose, it should be less than 140, but it's actually 210. So we see that this person has an excess amount of blood glucose, right?
Yeah.
So, okay.
Researchers detected glucose in the individual's urine and meat fibers and fat in the stool. Biopsy of the small intestine confirmed the presence of bile and the pH of 4, so it's acidic.
Okay.
Researchers also observed increased level of bacteria in the large intestine and osmotic diarrhea. Okay. The biopsy of the small intestine showed a non-pathological structure. A biopsy of the pancreas showed inflammation. Researchers suggested treatment with supplemental pancreatic enzymes. So now they're showing the detecting the glucose. They found bile and in their small intestine, and they saw increased bacteria and osmotic diarrhea in the large intestine.
What do you think osmotic diarrhea is referring to?
All I can think of is like osmotic pressure, like going solutes going in to something. He has a lot of guessing high osmolarity.
Yeah, so osmotic diarrhea, when your small intestine doesn't get to absorb as much of the water, and therefore you know, release it as a waste product.
Yeah,
And um, so it's a problem with reabsorbing the water in your
yeah,
face. And that yeah, okay.
okay. So said the pancreas showed inflammation,
Yep. And so
s-
they suggested treatment with supplemental pancreatic
enzymes.
enzymes. Now I was gonna also ask you, is the small intestine environment like very acidic?
The stomach is acidic.
Uh-huh.
Uh, Small intestine is in like you have the jejunum, the ileum.
Yeah.
You have those.
The most important one, though, would be the duodenum, right?
Duodenum, yeah.
That's the first like 20% of it. Now,
Yeah.
after the stomach, chyme.
Yeah. Or
So
yeah, a little bit.
that's just the partially digested food that comes from your stomach that
Hmm.
enters the duodenum. And since it's coming from the stomach, it's going to have a low pH because of the
Hmm.
acidic aspect of the stomach. But that's when your pancreas secretes bicarbonate ions, right, into the duodenum to to neutralize the acidity,
Yeah.
right?
So it should be neutral. It shouldn't be acidic.
It'll be like maybe like a pH of like 6
Okay.
or something. So, but we're seeing pH of 4, so we want to see this as being abnormal here.
Okay.
And there is also a an enzyme, the oh, wait. All right, never mind.
Okay. Researchers' analysis of the individual pancreas showed that the inflammation was due to CB3 virus infection. Uh, this Saki-B virus is a non-enveloped positive sense single-stranded RNA enterovirus. CB3 enters target cells through the CAR receptor located in the intercalated disc and or the decay accelerating factor DAF, which is expressed in epithelial and endothelial cells. The CB3 genome has an open reading frame of approximately 7.5 kb, and the virus uses cellular autophagy to promote replication. If CB3 infection reaches the heart, it could result in arrhythmia due to dysfunction and depolarization and repolarization of the myocardial cells.
What do you think autophagy means?
Uh, Something that eats something.
So the phagy part means eat. You're right. The auto,
Auto is like you're continuously eating something like a, I don't know.
so it means the self.
Yeah. So you're eating yourself?
Huh? Yeah. So, okay. So this passage had a lot of like buzzwords and things that we want to kind of pay attention to. So right away, we see that a person, who's experiencing diarrhea has weight loss despite eating a lot of food. And they did a biopsy of his pancreas and small intestine. Um, and they did a glucose tolerance test, which is in table 1, and we see that this person seems to have a glucose tolerance, right? Because this person has way more blood glucose than he should have, right?
Yeah.
So that uh, is an issue. And then they also detected glucose in the individual's urine. That's also like a symptom of like diabetes. meaning that you are peeing out glucose. And we know from the nephron that glucose should be something that we reabsorb, right?
Yeah.
Okay. They also found meat fibers and fat in the stool. So that tells you that the person's digestive system is having a hard time breaking down meat and fat.
Yeah.
Like, and instead of it being reabsorbed by the body, uh, it comes out in the stool, right?
Hmm.
All right. And then when they did the biopsy of the small intestine, they found the presence of bile. Tell me about bile.
The fat, it's stored in the gallbladder. Uh, s-
What does it do?
Stores fat or
So it emulsifies fat.
emulsifies fats. Yeah.
So what that means is, let's say that we had a circular kind of like spherical type of like globule of fat or oil. Um, and it has this surface area, but bile can like kind of, let's say, cut it in half to get two of these. And
Hmm.
if you have more, you have more surface area, right?
Yeah.
If you have more surface area, it's easier to like solvate the fat or oil here, right?
Hmm.
So bile helps emulsify, fats, which make them easier to perhaps digest. Um, so okay. And the pH of 4, we know that that's abnormal. So this person could have an issue with producing the bicarbonate uh, to neutralize the acidic chyme, right?
Okay. Should we go through the questions?
Yeah. I just want to also say that we also found increased levels of bacteria in the large intestine and osmotic diarrhea. So that means that the person, in addition to not retaining the whatever glucose, meat fibers, fat, and that exiting the body, water is also exiting the body. And then they found that the small intestine was fine, but it's really something happening with the pancreas. And then they found out that this was because of the CV3 virus, and then they talk about non-enveloped positive sense single-stranded RNA virus.
Hmm.
And that it binds to CAR on the intercalated discs or the DAF, which is in epithelial and endothelial cells. And then there's the reading frame of that many bases. That's 7.5 thousand bases, right? Per KB. And yeah, it uses cellular autophagy to promote replication. And if it reaches the heart, it's gonna pretty much really mess that up. So, okay, let's try the questions here.
Okay, it says which hormone levels may be reduced in the individual described in the passage. So he has high blood glucose, which means you have high insulin.
Okay.
So I don't think insulin would be
Well, what does insulin do?
it reduces blood glucose when blood glucose is high.
Okay.
But then it says which hormone levels would be may be reduced in the individual.
Uh-huh.
If you're reducing insulin, it means you don't have high blood glucose.
Well, tell me again what insulin does.
It's released when you have high blood glucose to kind of
So that's when it's
lower.
That's when it's released, but what does it do?
It reduces blood glucose.
Okay. Now, if you end up having high blood glucose, what isn't doing its job?
Insulin.
Mhm.
So their insulin is reduced because you have high blood glucose.
Yep.
Okay.
So I think what you were thinking of was the circumstances that cause insulin to be released.
Yeah.
Because high blood glucose means insulin gets released, right?
Yeah.
But that's different from what insulin does, right? So insulin actually,
Yeah.
yep,
Reduces. Hmm.
yep.
But then I don't know what the other ones kind of are.
Yeah, so the secretin, I was taking a look at that before because that's the thing that um causes the pancreas to release the bicarbonate to neutralize the acidic chyme. So, yeah, that was something that I was thinking about when I saw it.
Okay. And then gastrin just released from G cells. And then cholecystokinin. I don't know what that is.
Oh, it's the one that's responsible for um the breakdown, I believe, of fats, but let me make sure here. Stimulates basically the pancreas to release digestive enzymes, stimulates the gallbladder contraction, stimulates intestinal
Yeah.
motility, insulin secretion, etc.
Okay. Oh, it isn't.
Oh, that's these answer choices don't make sense. Well, because you're saying the pancreas, and I mean the question stem doesn't say only in the pancreas. I know in the passage we talked about the pancreas and the small intestine, so
Yeah.
pretty weird, but whatever.
Okay. According to the passage, CB3 most likely disrupts the function of which structure in the heart? Okay. If we go back, it says if CB3 infection reaches the heart, it could result in arrhythmia due to dysfunction and depolarization and repolarization of the myocardial cells.
Huh?
When, when I feel like we're talking about depolarization, we're talking about like either like voltage gated or like ion channels. So I feel like I have to do something with ion channels.
Very good.
Endothelium, they talked about it here, but it's not really
Mhm.
talks about DAF.
Yep.
Which structures did researchers observe in the biopsy of the small intestine? So biopsy, small intestine. We had the bile and we had the pH. And then they also saw increased levels of bacteria. Oh no, this is the large intestine. The biopsy showed a non-pathological structure. I feel like it's referring to like the non-pathological structure part. And then if it's non-pathological,
Mhm.
it would, I'm guessing between either B or C. Guessing, I'm thinking like B.
So what are we looking for?
Saying what did they observe in the biopsy of the small intestine?
Are we
And
looking for
then
normal or abnormal?
abnormal.
Um,
Or-
what does it mean to be non-pathological?
I don't know.
We could use a card trick here. Contrast word, inflammation. Inflammation's bad. So maybe
So
non-pathological means it's normal.
yeah,
So
okay,
if something's pathological, it means it's not normal.
okay.
It means it's bad.
So if pathological means bad, non-pathological means this is normal.
Yep.
Okay.
Because again, if it was pathological, they would tell us like, it has some type of problem, right? Like for the pancreas, it's inflammation. But
Yeah.
so what is it normal to have flattened microvilli
No.
and a few blood vessels?
No.
Because, uh-huh, okay.
So what's Rugen?
So that's a good question. I actually looked that up. So it's apparently the folds within an organ, like the stomach.
Okay. All right. Microvilli, many small capillaries and lacteals. I don't know. Is it that one?
Uh-huh. Because your microvilli will be like, things like this, right?
Mm-hmm.
Like kind of like the brush border cells.
So is this one for D? It says infiltrating white blood cells. Does that mean you have too many white blood cells or
So that's saying that, so first it's saying thick epithelium, which is the outermost layer, right? And then
yeah,
it's saying infiltrating white blood cells. We, our blood cells should be in our like um blood, right?
yeah.
So if it's infiltrating into the epithelium, it means that it's coming out of where it's supposed to be.
Okay.
So, but you know, the microvilli you want to be aware of. So let me show you a picture here. So I'm gonna put it in the chat. So, so yeah, you essentially have a bunch of these, and above it, you see the microvilli. They are projections,
Yeah.
and it's almost like a brush, and that just increases the surface area of our intestines so that things can get, more so digested, and they can appear um in this type of in something like this here's the second picture. Yep. So you see how, so this is the lumen with the villi, and the villi are already these projections, and
Yeah.
they're coated on the outside with the microvilli, right? With cells that have the microvilli.
I've seen this picture before, but I didn't really know what the function of them were.
So does it make sense that we want as much surface area as possible?
Yeah.
So all of these microvilli, in addition to the regular villi, enable the food to get, properly digested and properly absorbed back into the blood through the microvilli.
Hmm.
It's like if you have like a water bottle and like the brush that you put it in,
Yeah.
that's kind of like that. So the hairs of the brush have that surface area that it can pick up, parts of like the water and stuff like that for the water bottle
Okay.
example. But this is for absorbing, nutrients and stuff.
And
Now,
it's in the small intestine.
yes. Now, inside of what I sent you is going to be the lacteal. So, yeah, so here, like, and here you'll have um uh so it's not exactly like a blood. I mean, sure, there is blood vessels there. Here, I'll just get a better picture here, but you're gonna have a thing called a lacteal, which is part of the lymphatic system, and that's because the fats don't get absorbed readily into the blood. So they get absorbed by the lacteals, which enable the lipid stuff, fats, to get absorbed into our lymphatic system. Here, here's
Okay.
a good picture. Yep.
Mm.
Yeah, this is actually the best picture. So we see everything here. So we see our overall villus, or right, villi, and inside we see the small ones are the microvilli, and we also have the columnar epithelial cells. They're called that just because they're long, and
Yeah.
tall, like a column. And then you have your capillaries, right here. And then the green thing here is your lacteal, which is part of the lymphatic vessel, or the lymph system. So this is how fats, so like water soluble things can just get absorbed into the blood, into the capillaries, but hydrophobic things like, fats and oils, they get absorbed by the lacteal and go through the lymphatic system.
Okay.
So that's why we see microvilli, we see a lot of capillaries at the surface, and we also have lacteals.
Okay,
Yep.
makes sense.
Mhm. Yep. Many small capillaries and lacteals.
Which type of enzymes did researchers most likely suggest the individual be treated with? Um, the researchers suggested treatment with supplemental pancreatic enzymes. Um,
So what what would pancreatic enzymes generally do?
and um, the release is glucagon, but
Oh,
that doesn't really, yeah,
think about the
that
ones
doesn't.
uh that have something to do with, you know, the digestive system.
Hmm.
So
I'm not really sure. Like, what do you mean?
sure. So okay. Meat, fibers, and fat in the stool means that they're not getting properly broken down, right?
Yeah.
And we know that there's an issue of the pancreas,
Hmm.
which, you know, likely is unable to create some of these enzymes which is why they're treating the patient with supplemental pancreatic enzymes. So if the person's pancreas is not working properly and you see, you know, undigested meat and fat in the stool, pancreatic
You
enzyme,
would
yeah.
you would want something to bring them together,
Uh, what do you mean?
or like if they're not being digested properly,
Yep.
you would want something to digest them.
Hmm. So now is digestion bringing things together or is it something else?
It's breaking stuff apart.
Good.
So it's either hydrolase or lyase, but hydrolase uses water to break something apart.
Uh-huh.
Here, we're not talking so much about water or it says individual's urine. Um,
Um, so okay, so meat is a type of is protein, right?
yeah,
Protein is composed of
fats
proteins are
or
fats,
amino acids.
amino acids, right?
Yeah,
So how do you go from amino acids to like a protein? So like a protein is multiple amino acids, right?
yeah.
That are all bound. So how are they bound?
Covalent bonds.
What type?
So what type of covalent bonds? Uh, um,
So just think about two amino acids.
like disulfide bonds. So
So
like cysteine. I don't know.
so okay, so would you be able to if
Peptide bonds.
yeah, which one?
peptide or
Yeah, because if
yeah,
you so yeah, you want to be able to draw an amino acid.
yeah,
So it'll look like this.
the N terminus, the C terminus.
Yep, an R side chain and then a carboxylic acid group, right?
Yeah,
And then we have another one here. So in order for these to bond, what happens?
condensation reaction,
All right. In other words,
dehydration,
dehydration what?
dehydration reaction, where
So
you're releasing H2O.
it's a word that means so the dehydration part already accounts for releasing H2O, but what's the word that means you're bringing things together, combining?
Ligase.
Oh, I just mean like the general word starts with an S for
They
combining
say
things.
like combining these two.
Yeah. It starts with an
Synthesis.
yes. So dehydration synthesis means you're combining these two amino acids with the loss of water, right?
Yeah.
And then that produces your amide bond, right?
Mm-hmm.
Which is the peptide bond, right?
Yeah.
Okay. And so if they synthesize through the loss of water, if you want to break them, what must you do?
Use water.
Okay. Now that's going to be the same also for the fats. Because remember, a fat like a triglyceride is you take a glycerol molecule like this, you take a fatty, like three fatty acids, and you bind them together through dehydration synthesis also.
Mm-hmm.
So that's why hydrolysis would break the proteins, the peptide bonds of the protein,
Oh, okay.
as well as the uh, ester bonds in fat. So,
Okay.
yeah. So,
Makes sense.
you're right to eliminate B and D because B would bring things together, right?
Mm-hmm.
And D is just redox.
Yeah.
And then A and C are both breaking things down. But A is breaking things down you with the addition with through hydrolysis.
Yeah.
Um, and that's typically going to involve like the peptide bonds or the fatty ester bonds, things like that. Whereas a
Mm-hmm.
lyase would be more so breaking like a carbon double bond,
Yeah.
stuff like that.
Okay. So it'd be
Pretty cool because like this passage seems to be like very clinically relevant, right?
yeah. It's combining a lot of different
Uh-huh.
stuff. Yeah.
Yeah, and it's showing you like, if you find urine and meat fiber and oh sorry, glucose in the urine and meat fiber and fats in the stool, it makes
Mm-hmm.
you think like, okay, like these are actual symptoms and what could be the cause this person's doing all that. So
Yeah.
pretty cool. Oh, and then
Mm-hmm.
I knew they were going to ask this. Um, right.
Ask about
Yeah.
this.
Yeah. Except this is a little bit has like an extra kind of step.
Okay. It says what is the approximate molecular weight of the polyprotein translated by the virus? I was talking about, so the open reading frame is 7.5 KB. Is it talking about like the amino acids and then like the 110 DA is one amino acid?
So that's going to play a role, but
Says
there is an extra step here. So what does the 7.5 KB represent?
the open reading frame.
So
And then it's saying, oh, translated.
so so
Or
so what does it represent? 7.5 kilobases.
the genome.
Okay. So nucleotides, right?
Nucleotides, yeah.
So and now how can we go from nucleotides to amino acids? Like when you're translating, right? Is it
Yeah.
like one nucleotide translates into one amino acid or
Thought it was three nucleotides for one. Okay.
yep.
So then one protein is 110 Daltons
An amino acid, okay.
per one amino acid. Mhm.
So what are you doing in the math?
Now I'm just writing down three nucleotides for one protein, and then one Dalton is for one amino acids, but I'm not
Oh,
really sure what
oh
this
yeah.
question,
No, you're
like
doing
how
it
to
right.
go about it.
You're doing it right. So it's just going to be three nucleotides per amino acid.
Yeah.
So 7.5, I'm going to just write instead of the KB, right? I'm going to write E3, B for the bases, okay?
Yeah.
Right? And then for three bases, you get one amino acid, right?
Yeah.
And for each amino acid, you get 110
Daltons.
Daltons. And I'm just going to write that as 1E2 Daltons. Or you know what I can do? So 110 Daltons is 0.1 kilodaltons, right?
Yeah.
And so I'm just going to write this as 1E minus 1 kilodaltons, right?
Hmm.
Okay.
So 7.5 times 10 to the 3 times 1 times 10 to the negative 1. On the top, you would have 7.5 times 10 to the 2 over 3. Then it will be like, this would be in Daltons. Oh no, this would be in kilodaltons. So the bases will cancel out. 7.5 over 3 would be 2.5 times 10 to the 2 And then this is Daltons. Oh, no, kilodaltons. And then you can just move the decimal over to the right twice. Got this.
Tell me what you said again. You said you had what?
So you have 7.5 times 10 to the 3 bases. The bases cancel out. And then over here you have 1 times 10 to the negative 1 kilodalton.
Mhm.
So if you multiply these, you would get 7.5 times 10 to the 2 over
Oh,
3.
you mean so the three goes into this 2.5 times, right?
Yeah,
2.5 E3 times 1 E negative 1 gives us 2.5 E what?
to the 2.
Which is just moving the decimal place to the right twice.
Yeah.
So wouldn't that be 250?
If you move it to the right.
2.5?
Oh, yeah, it is 250. Yeah.
Huh? And why do you think, so the answer should be this, right?
Yeah, yeah.
Why do you think this says 270 rather than 250?
Because we
Mhm.
got from 110 to 0.1.
Yep.
We
So,
went down, so our answer should be a little higher.
exactly.
Okay.
But be very careful with the math.
Yeah, yeah, yeah,
You
yeah.
want to get a, yeah. And
Okay.
you see how I said, it's an extra step
Mm-hmm. This
because
is the 3 nucleotides for 1 amino acid.
yep,
And then you do the 110 daltons. Okay.
exactly, yeah.
All right.
So yeah, so it's cool because like they already know that we know how to do questions that say like, okay, this is this many amino acids, and then figure out, the weight of it. So they're just putting in another step there. So that's something to expect from the MCAT. There's usually going to be rarely going to be that straightforward. Like it'll be usually some multi-step kind of thing.
Okay. Which process is most likely observed in CB3 infected cells?
Oh, this could be kind of annoying because it could be just like a you know it or don't, but maybe we can do process of elimination.
So it says the virus is a non-envelope positive sense single-stranded RNA.
Okay.
So it wouldn't be in DNA.
Well, they're talking about what in the question stem?
Says which process is observed in CB3 infected cells?
So are we talking about the CB3 virus, or are we talking about
Oh, we're talking about the CB3 infection.
yeah? So the
Okay.
cell, right?
Yeah. So it says production of high levels of caspases. Is it talking about something with the heart?
Um, are you asking if Caspases have something to do with the heart, or the question stem?
I don't know where to look for in like the passage. Says which the following is observed in CB3 infected cells.
So let's think
Are they
of
talking about these target cells?
well, yeah, because it said CB3 infected cells, right?
Okay.
So it'd have to be cells that are infected with CB3.
Hmm.
So now, what does CB3 do in infected cells?
What was the question?
So, like, what does CB3 do inside, an infected cell? Like, what is the passage?
Replicate
Sure. Everything does that, but what does it do, like, to the infected cell? So, what does it say in the passage?
the inflammation was caused by the virus infection.
No, no. So, when the CB3 enters or infects a cell, what does it do to that cell?
It says the virus uses cellular autophagy to promote replication.
And we talked about autophagy being what?
Self, you're eating yourself.
So
So it'd be the replication of intracellular organs. All right.
if you're eating yourself, why would you have, replication means you're making more of something, right? So if you're making more organelles, you wouldn't really be eating yourself, right?
Yeah.
So C we can eliminate.
Would you be able to eliminate D2 because it talks about replication?
Yep.
Uh, I don't know what caspases are.
Okay.
So I think it would make new cell membrane
So why did we eliminate C and D?
because it talks about replicating and making something,
All right. And what's B talking about?
making something.
Right.
Yeah.
So this is an occasion where we don't know what caspases do. So what do, right? Well, we know from the passage that the virus causes the infected cells to eat themselves. So we eliminate things that talk
Have to
about,
do with the replication.
yep.
Okay.
And that means that since we know B, C, and must be wrong, whatever A is must be right.
Okay.
So see the logic there?
Yeah.
Because you're never going to know everything.
Yeah.
You want to be prepared to be able to deal with that uncertainty. And now
Mm,
they said that
awesome.
caspases um cause cell death. So now you could just add that to your knowledge. Maybe you could even make like an Anki card for it. So in the future, you'll know that caspases do cause cell death, apoptosis.
Okay.
But
All right.
yeah,
Which sequence of events does the virus most likely use to replicate in host cells?
yeah, I knew they were gonna ask this when they said positive sense.
Single-stranded RNA enterovirus.
Yeah. Um,
Says
hmm?
generation of DNA from the viral genome followed by DNA transcription and RNA translation.
Do you remember what positive sense and negative sense what were?
Isn't positive sense like replicate? Um, Negative sense is like replicating into yourself, kind of.
So
Or complementary or something?
So your positive sense RNA is can be directly translated into proteins, meaning that it's the same sequence as the mRNA,
Okay.
but negative sense RNA is complementary to the mRNA. So that has to first be converted to positive sense RNA by RNA polymerase before it can be translated into proteins.
Okay.
So,
So positive sense RNA is can be directly translated to proteins, but negative sense RNA is complementary, so it needs DNA polymerase to be translated to positive sense.
yep, RNA
Or
polymerase.
RNA polymerase, yeah.
Mhm.
Okay.
To make the positive sense one, and then the positive sense one gets translated. Yep.
Yeah. This one's talking about DNA. I don't think it's right.
Um, okay, yeah, that would be like a reverse transcriptase, right?
Yeah.
Retrovirus.
Integration of the viral genome in the host cell genome, followed by replication within the host cell. Viral genome. I don't think that's right.
What?
It doesn't talk about like the RNA or the positive sense or any of that.
Well, what would the host cell genome be made of? You don't need the passage for that. The host cell is who the host is who?
The protein?
So like if a virus infects a host, the host is the person who's infected with the virus, right?
Yeah.
So what is the host cell genome made of?
DNA?
Okay. What's the viral genome made of?
RNA?
So can you integrate RNA into DNA?
Yes.
You can. You could just mix, you could have a strand where you have some DNA and some RNA and they're all part of the same strand.
Oh, no, no.
Okay.
You need, well, you need the RNA, like the positive sense
Um,
to directly to the proteins,
yeah, but
but not for DNA.
but you see how this is saying you're integrating the viral genome into the host cell genome?
Yeah.
So like that means that you're taking the host cell genome and you're mixing in the viral genome. But that can't be because the viral genome is RNA and the host cell genome is DNA,
DNA.
so they can't be integrated that way.
Okay.
So you want to be careful whenever you say like they never talk about this in the passage because that'll be a lot of things,
Mm-hmm.
right? Okay.
Retrotranscription of the viral genome and integration in the host cell genome, followed by replication within the host cell. Isn't it kind of the same deal as B, where it talks about host cell, which is DNA?
Now, this is what's retrotranscription?
Oh, it's from RNA to DNA?
Huh? So this could theoretically work, right? The viral genome, if that's RNA, it gets reverse transcribed into DNA, and then it can be integrated into the host cell genome, right?
Oh yeah, yeah, yeah.
So that can work, but they're talking about a virus that is not a retrovirus, right?
Mm-hmm.
So, C, while it could be something that happens, it only happens if you're a retrovirus,
Yeah.
meaning you need to have reverse transcriptase.
What do you mean like they don't talk about retrovirus? What it like in here it says
So a retrovirus is a virus like HIV,
yeah.
which has an enzyme called reverse transcriptase, which means it can do the reverse of transcription. It can take RNA and reverse transcribe that into DNA. But this
Mm-hmm.
is only true for retroviruses. So they never said that this was a retrovirus, CB3.
Okay. So if this didn't say if this said retro, then C would be right.
Yeah,
Okay. So what's enterovirus? Just a regular virus or
I think it's a virus that infects the uh, digestive tract, but let me make sure. Yep, they're viruses that get transmitted through the intestines.
okay.
But I was just thinking a GI doctor is a gastroenterologist.
Enterologist.
So, entero. You always want to use, whenever you don't know
Okay.
a word, you always want to use things that you know.
Okay. And it says direct RNA translation of the viral genome and synthesis of negative strand RNA, followed by replication of positive strand RNA. So is this talking about when it says the synthesis of negative strand RNA? Is it talking about negative strand RNA using the RNA polymerase to become positive strands?
Oh, so this is just saying that we already have the positive sense, and that's why you can directly translate the RNA of the viral
Yeah.
genome. But you can also make the negative sense um, RNA that can be uh, that can help make the positive strand another version of the positive strand to get translated, continuously. But yeah, positive sense, direct translation. That's why this will be that.
Okay. That makes sense. Okay.
All right,
Good.
that's pretty cool. So,
That
uh,
was useful. I
yeah,
needed something like that
yep,
to see what to like review and stuff.
yeah,
Because
yeah,
when you're by yourself, it's a little bit like,
mhm,
oh, I know this, but it's not like how everything
yeah,
connects.
yeah. So awesome, awesome. So yeah, that was a good passage. It had a lot of those nice buzzwords and things like that. So I think what you're gonna do is, are you gonna take a practice test soon?
Yeah, Friday's my next one. I'm taking LTS 5.
Got it.
Um,
And okay, awesome. Let's see. Should be 132 for today. That's going to be your score on these sections.
yeah,
Mm-hmm.
we'll see. Okay, let me know when you get it
Mm-hmm.
on Zell.
Yep, I got it.
Okay. Sounds good. All
Awesome.
right.
So
Okay.
good luck on the
Thank
test,
you.
and I will see you. You'll probably make some appointment.
Yeah, either probably Tuesday, because yeah, probably either next Tuesday or Monday I'll make one.
Got it. Yeah, do it.